Well, thank you so much for coming today. I'm excited to share my work with you, some ideas about how to use biotechnology for carbon dioxide removal, a topic I think is really exciting and cool. Um, so I've been working on this roadmap for using biotech in CDR, and I'll tell you about the findings of the first part of that roadmap, which focus on industrial carbon removal using aqueous solutions. And before I go ahead, I just want to say thank you to Additional Ventures for funding this work, and also to the Innovative Genomics Institute at UC Berkeley for hosting me as an independent fellow as I carried out this work. So we'll start by talking about who I am, who we are, and what this report is. Then we'll go to talk about the core technologies that we're dealing with and how biocatalysts can help to improve them. Then we'll talk about ideas we'll break all of that down, and we'll encounter two themes for the future work that will be necessary in my view, and we'll on that journey discover 14 specific fundable projects, and I'll give a little more detail on what that means. So I'm a co-founder of Homeworld Collective, along with my dear friend and colleague Dan Goodwin. We exist to help practitioners in climate biotech find good problems to work on, find community, and find work and get working on problems in climate. And the reason that we think an organization needs to exist on behalf of the climate biotech community is because historically, funding and effort in biotech has gone pretty much dominantly to medicine, even though there are enormous opportunities and potential for biotech in climate and sustainability. Luckily, that's changing, and we're supporting people as they make that transition, as they discover new problems and map out new problems in this space to help climate biotech gain the cohesion and momentum that medical biotech does. And I just want to point out that throughout this webinar, we'll be highlighting some potential projects, and we'll be highlighting some potential projects that we're hoping to fund, and we'll be calling them fundable projects. And what this means to us is that, given that this is a roadmap, the goal of this work is to identify problems on the frontier that need to be worked on. And so we want to help people work on those, and we want to help those projects get funded. And so if you see a project that you think you're interested in, please reach out to me. So I co-authored this work with Colin McCormick, who's been a wonderful mentor on this project for me. And if you want to look at the written version of our roadmap, go to homeworld.bio/research, and you can check it out. It's hosted on a platform called PubPub, and you'll be able to comment if you have things to say, and that's why we chose that platform. So please engage with it if you want to get into those nitty-gritty technical details. Throughout this webinar, I'll try to stay out of the technical weeds as much as I can, and give us some actionable takeaways. So why are we here talking about CDR, carbon dioxide removal? Well, as many of us know, the National Academies of Sciences, the IPCC, have told us that we'll need anywhere between 1 and 10 gigatons per year of carbon dioxide removal by mid-century in order to meet our climate goals as we decarbonize as a society. And this work, CDR and also decarbonization, is really our work. It's the work of our generation. We need to make these changes over the coming decades. And so that's why we're talking about it. And I've included this obligatory image from the National Academies of Sciences, very frequently used, and is a great illustration of just how much work needs to be done. Now, I'll be mapping opportunities to contribute to CDR specifically for one discipline, which is biotech. And why am I doing that? Well, the goal is to connect specific people with a specific skill set to problems. And this needs to be done across all skill sets. But it's important that we speak to people so that individuals can get moving. So we start by breaking down the problem, thinking backward from CDR, breaking it into its constraints, and then thinking forward from the motivated community. What are the tools, skills, and knowledge that we have? And how can we map those to address some of the constraints? Of course, not all of the constraints in CDR can be addressed by biotech, but some of them can be. And so we're trying to discover those to help folks in biotech orient themselves. Now, why specifically biotech? Well, the 21st century is heralded as the century of biology, and we know that it also has to be the century of decarbonization. And biotech has been undergoing a revolution in the last few decades. And this figure helps to illustrate that. This is a measure of the cost of DNA sequencing over time. And note the log scale on the left. DNA sequencing technology has been improving faster than Moore's law for decades now. And that's representative of other aspects of biotechnology, including DNA synthesis, protein engineering, genome engineering, and also our foundational knowledge of biology, which we use to do biotechnology. And I think there are a lot of opportunities for biotech to contribute to carbon removal. And we'll see that throughout this talk and throughout the rest of the roadmap. So this roadmap is focused specifically on road mapping opportunities for biotech in neglected areas of CDR. So far, the main focus for biotech in CDR has been on nature-based solutions like soil carbon, forestry, cultivating biomass in the ocean and sinking it. These are worth working on, but they're not the focus for us. Our goal was to look at how biotech can be applied to the engineered and abiotic methods, which so far have been more neglected. And we bin those into three categories. The first is industrial carbon removal using aqueous solutions, which is direct air capture, direct ocean capture, which we'll talk more about those later on. Also bioenergy carbon capture and storage, which is burning biomass and collecting it using point source capture. But because that carbon came from biomass, which came from the air, that constitutes carbon removal. We're not going to talk about BECCS and point source capture today in this webinar, but it is discussed in the written roadmap. The second bin is geochemical carbon removal using minerals and their alkalinity to remove and store carbon. And the third bin is biomass. Can we engineer bigger sources of biomass and biomass that is most appropriate for carbon removal and storage? So before we get into it, I just want to go over a couple core concepts, which are direct air capture, direct ocean capture, which are the two CDR technologies we'll be talking about, and also protein engineering, which is the primary tool that we'll be talking about. So direct air capture is a technology that blows air over an absorptive material that holds on to carbon dioxide and then removes that carbon dioxide to collect it in a concentrated form and then store it. And this this image by carbon engineering of a rendering of what a megaton scale DAC facility would look like has sort of become emblematic of direct air capture. Now, the cost of direct air capture using today's technology when scaled is expected to be anywhere from $100 to $1,000 a ton. And the core constraints on that cost are the size of the air canter, or the air contactor, this giant apparatus that is used to pull dilute carbon dioxide from air, and also the energy used to pull the carbon dioxide off the absorptive material to then take it and store it. Now that energy primarily comes, like I said, from CO2 regeneration, which I'll just define here because we're going to be using this term. CO2 regeneration just means removing CO2 from solution to collect it and store it. Now that energy is about, for current technology, between 4 and 20 gigajoules per ton of CO2 removed. And 4 gigajoules per ton at 10 gigatons a year, which we're told we are going to need, would be 6% of the world's energy usage just for direct air capture. So we're talking about an enormous amount of energy when scaling these technologies. The other technology we'll talk about today is direct ocean capture, which is similar to DAC in the sense that it's processing a fluid to try to remove CO2 from it. In this case, the fluid is water, seawater, which contains carbon that has come out of the atmosphere. We process that water in this facility. We pull the carbon dioxide out. We let the water out the other side back into the ocean, and there it equilibrates again with the atmosphere, and we've done net carbon removal. The cost of DOC using current technology at scale is estimated to be anywhere from $500 to $2,000 a ton. And the main sources of that cost are the electrodialyzer unit used to create a pH swing to accelerate the CO2 coming out of the water, and also the energy used to produce that pH swing. The energy for DOC is estimated to be around 3 gigajoules per ton using today's technology. Now, as we'll discover throughout the webinar, there's good reason to think that biocatalysts can reduce both the cost and energy of both of these technologies, which are part of the core challenges of improving them. Now, let's talk about proteins and protein engineering for a sec, because we're going to be using proteins to improve DAC and DOC. So proteins are the machines that do the magic of biology. And here's an image of a very cool protein complex, the flagellar motor. So on the top here, we have real images. Whoops. On the top here, we have real images captured using cryo-electron microscopy of this protein structure. On the bottom, we have cartoon depictions of the structure. This is a tiny machine. I should have a scale bar here. This would be this would probably be 100 microns across or something. And it would involve hundreds of individual protein molecules. It's got an enzyme on it that catalyzes a reaction and uses the energy that comes from that reaction to turn this wheel, which causes a flagellum to swing around, which is sort of the tail that is on some cells to help them swim around. So proteins are really magical. I mean, when light hits your eye, it's a protein that receives the photon to tell your eye that there's light there. When you eat food, it's a protein, an enzyme that will digest your food into its constituent parts, and then other enzymes will build those parts into you. So they really can do a lot. And now proteins are determined by DNA sequence. It turns out that you can, if you want a specific protein to be created, all you have to do is understand the sequence that produces that protein, and then engineer that into a cell, get the cell to produce that specific protein, and you have your protein. That's a bit of a trivialization, but at the core, that's the concept. And now protein engineering is really the next revolution in biotech, and that's happening now. We've had DNA sequencing, which allowed us to gain a window into how biology works. We have DNA synthesis, which now lets us build biology, and people are maybe familiar with the term synthetic biology. And now that we have those things, we can apply our knowledge, our ability to synthesize, as well as emerging tools like artificial intelligence, to design proteins. And here on the bottom right are a collection of protein structures that were designed using artificial intelligence. So how can proteins improve DAC and DOC? Well, let's talk about how DAC works. We start with an air contactor. We have fans blowing air over a solvent. In this case, in this talk, we're just talking about direct air capture using aqueous solvents, so not solid sorbents today. The CO2 goes into the solvent. It's carried in the loaded solvent over to a regeneration unit where it's subjected to heat or pH swing. The CO2 comes out and then is taken away to be compressed and stored. Then that lean solvent, which no longer contains the CO2, goes back into the air contactor where it can be in contact with carbon dioxide again and it loops like that to pull carbon out of the atmosphere. Now, fast absorption of CO2 into the solvent minimizes the size of that air contactor, which is a leading component of cost. So you want that absorption to be as fast as possible. And low energy regeneration minimizes the energy needs. You want to make it easy to get the CO2 out of the solvent once you've captured it. However, in practice, optimizing one of these factors trades off the other. It's hard to get fast absorption and low energy regeneration, and doing that is one of the core challenges of direct air capture. Now, the enzyme carbonic anhydrase evolved specifically to enable low energy carbon dioxide exchange. Here's a diagram of carbonic anhydrase. And variants of this protein exist all across every organism that we're aware of. In humans, there are like 8 different carbonic anhydrases, and they facilitate the exchange of CO2 between our cells and our blood, and also our lungs and the air. And carbonic anhydrases are also present in bacteria. This is a cyanobacteria that has to absorb CO2 quickly and easily in order to photosynthesize and turn that CO2 into biological material. Now, the enzyme catalyzes a specific reaction in the absorption of CO2, which is the conversion between carbon dioxide and bicarbonates. That is the rate-limiting reaction. All the other components of the process are actually pretty fast. So at a high level, we can start to see how carbonic anhydrase can give a resolution to the trade-off between absorption rate and regeneration energy. So the solvents that we currently use to capture carbon dioxide in DAC are strong bases. So that's like an example of sodium hydroxide. These have a very fast absorption rate because they're very reactive with CO2, which means that they have a relatively smaller contactor size, still gigantic, but relatively smaller. And that's good because that minimizes cost. However, because those strong bases hold on to CO2 so strongly, they have a high regeneration energy. It's hard to rip CO2 out of a strong base. And so that means we need a lot of energy in order to do that, and that drives high energy costs, so that's bad. And we alternatively could use a weak base as a solvent. That has a low regeneration energy because it doesn't hold on to the CO2 as strongly, which means it has low energy usage, but it also has a low absorption rate, specifically because it isn't as reactive with CO2. And that means it's going to have a relatively larger contactor size, which is bad. Now, if we use the enzyme carbonic anhydrase to accelerate the absorption rate, which is what it has evolved to do, that can give us a fast absorption rate in weak base, along with that low energy regeneration energy that the weak base already gives us, to allow us to have a relatively smaller contactor size and lower energy use. And that means we can co-optimize the 2 main cost factors in direct air capture. So our goal is going to be to enable efficient and economical use of carbonic anhydrase in a weak base capture solvent for direct air capture. So has carbonic anhydrase been used in DAC before? Well, for DAC, there actually have been no analyses or publications devoted to using carbonic anhydrase in DAC. But there has been substantial work for using carbonic anhydrase in point source capture. There's been recently, in 2022, commercialization of a point source capture technology based on carbonic anhydrase as a collaboration between the heavy industry company Sapem and the enzyme company Novozymes, which is very exciting and encouraging. And some numbers from a techno-economic analysis tell us that basically using CA in point source capture can give 41% energy reduction and 31% cost reduction relative to the conventional approach. And oh yeah, I wanted to define techno-economic analysis just so we're all on the same page. We'll revisit this concept a few times. Techno-economic analysis is modeling of the economics of a technology to assess its feasibility and to guide future research and tell us what the expensive parts are, how things can be co-optimized, and what targets should be for different components of a technology. Now, given that there is substantial work and promising work for using carbonic anhydrase in point source capture, that is encouraging for its use in direct air capture. Direct air capture is different because it's pulling CO2 out of air, which is at a much lower concentration. And so the solvents we use and the process design is a little bit different. But nonetheless, this is encouraging. And from my perspective, the reason that we haven't been using CA and DAC, and we have been in point source capture, is really just because DAC is newer. And the communities that are good at using these things, the biotech community and the DAC community, are just not very integrated. And so that's going to be a theme throughout this webinar. Now let's also talk about direct ocean capture. Shifting gears now, we're pulling CO2 out of seawater. The way this works is seawater goes into the dock facility where it's subjected to vacuum pressure. And so because there is a difference in partial pressures between CO2 in the vacuum and CO2 in the seawater, the CO2 comes out of the seawater, and we can collect it and store it. Now, the current technologies for dock also use acid produced by an electrolyzer to accelerate the rate of CO2 coming out of the solution, or coming out of the seawater. And then on its way out, we put base back into the seawater to balance that pH, and then the CO2 depleted seawater will equilibrate with the atmosphere and accomplish carbon removal. Now, it turns out that the acidification that we're using to help the CO2 come out of the seawater is actually only there to boost the kinetics, of the rate of it coming out. It's not necessary in principle thermodynamically. The vacuum pressure is enough to pull that CO2 out. And so really we're spending a bulk of the energy in dock to accelerate a reaction that an enzyme already catalyzes. And so that's going to be the crux of how carbonic anhydrase can help in dock. So if we use a biocatalyst, the seawater goes into the facility, it's exposed to the biocatalyst under vacuum pressure. CO2 comes out of the solution, and then it goes back into the seawater. And we can potentially, if we get a really high-performing catalytic process, obviate the electrolysis component of dock, and if not completely obviate it, at least reduce the amount of acidification, which would substantially reduce the amount of energy used. In the best case, the energy reduction could be up to 25% of the total cost, or up to 10% of the total cost just from removing that electrodialyzer unit. And this is just based on the contribution of those technologies to total cost by techno-economic analysis today. So our goal is going to be to enable efficient and economic use of a biocatalyst within the CO2 regeneration apparatus in dock. Has carbonic anhydrase been used in dock before? Well, no. There is one company that's beginning to pursue it. I'm not sure if they want me to share publicly which company they are. There are no academic publications on this topic and no modeling analyses. And so those will be issues that we'll be hoping to address. Now, as we talk about how to pursue biocatalysts in DAC and dock, there will be two themes that we'll encounter, and I want to stress these. The first is that we need clear engineering targets to orient R&D for using biocatalysts in CDR. The second is that protein engineers need to work with materials engineers, and process engineers. Currently, those communities are siloed, and but all of their skills are relevant to this problem. So we'll start by talking about biocatalysts in direct ocean capture, dock, because that's conceptually the simplest. So we've already encountered a fundable project. Again, this is a project that we think needs to be done and needs to be funded. And so we want to help people work on making these projects happen. So this project, the first project we encountered is that we need a target for catalyst performance in dock. There are no analyses. So specifically, What are the minimum and ideal catalytic rates and materials costs for replacing acidification with catalysis in DOC? And having those benchmarks and targets will allow us to pursue them. Now for DOC, digging into the details, the catalysts have to be immobilized within hollow fiber membranes. Hollow fiber membranes are these thin membranes. They can be about 200 microns. They can also be a bit thicker, up to a couple millimeters. That fluids can pass through. In this case, seawater can pass through this thin tube, and it encounters a selective layer that allows carbon dioxide to pass through, but not water. And so the water will go through. It in the DOC facility, it's subjected to vacuum pressure, which causes the CO2 to come out, and then the water goes back out into the ocean. And so if we want to expose the seawater to a catalyst, we need to immobilize that catalyst on the inner wall of the hollow fiber membrane. So that's our challenge. And people have done that before. The specific challenges we're going to encounter using catalysts in hollow fiber membranes for DOC really come from the membrane. The membrane lifetime and its permeability limit the utility of biocatalysts in DOC. And so the first component of that is its lifetime. The hollow fiber membrane is an expensive component of DOC, and it has a lifetime of about 5 years. And so the catalyst has to last 5 years or be capable of being refreshed within the hollow fiber membrane without replacing the membrane, so that the catalysts aren't decreasing the lifetime of the membrane. And that's a tall ask from a catalyst to last 5 years and be stable. Now, also the membrane permeability is going to saturate before the catalytic rate. So it turns out that because this catalyst is so fast, it can liberate carbon dioxide from solution faster than the membrane can actually allow carbon dioxide to move through at the small difference in partial pressures that you have between a vacuum and the dilute dissolved carbon in seawater. And so these facts together mean that the stability of the catalyst, making it last those 5 years or be refreshable, is more important than its total activity, at least for the current design for direct ocean capture. And that indicates that likely if we want to use a catalyst, we probably want to use a chemical mimic of carbonic anhydrase, like a zinc cyclone or one of many other small molecules that have been designed to catalyze the same reaction. Even though these molecules are orders of magnitude less efficient than carbonic anhydrase, they are more stable. So here we've discovered a few fundable projects. So on the theme of protein engineers, or in this case, molecular engineers needing to work with materials and process engineers, we have, oh, actually, that is, okay. This is not a TEA, this is an engineering project. Can we make hollow fiber membranes that contain an immobilized biocatalyst that is stable or replaceable over 5 years? Because that's what we need to do to enable the use of the biocatalyst in DOC. We also discovered a couple fundable projects related to improving the membrane permeability to match the catalyst efficiency so that both of them can perform optimally together. The first is an engineering project. Can we develop a higher permeability hollow fiber membrane that can fully leverage the efficient catalyst, the full efficiency of the catalyst? And that's for protein and molecular engineers working with process engineers. The other is an analysis. Could we leverage the full catalytic rate of carbonic anhydrase by using a smaller quantity of a more costly but higher permeability membrane material? And this would be considering, in the context of a very cheap catalyst of this reaction, can we just use a whole different membrane material? And that's an exercise in giving us engineering targets. Now let's talk about the possibilities for biocatalysis in DAC. So the first question that I encountered when I started looking into this is whether the carbonic anhydrase should be free or immobilized in DAC This is an ongoing discussion in the point source literature, which I'll dig into a bit now. So if you use free carbonic anhydrase, that means the carbonic anhydrase is just dissolved in solution in your solvent. And so in the air contactor, the catalyst is there with the solvent. It moves with the solvent into the regeneration unit, where it's subjected to heat or pH swing. Then it goes back into the air contactor again to come in contact with CO2. Now, this process of being subjected to heat or pH swing in the regeneration unit is a huge change in physical conditions compared to the contactor. And that means that the enzyme is subject to a swing of extreme conditions, which is difficult for its stability and can destabilize it. And that's the core challenge of free CA. Now, using immobilized CA, that means your CA is attached to a solid support in the contactor. And so it's only present in the contactor. It doesn't go into the regeneration unit. And so it isn't subjected to that other set of conditions, which protects it. And immobilization is trending now in the point source capture research, basically because it can reduce the cost associated with using the enzyme. It reduces the total amount of enzyme that you need, because the enzyme is only present in one place rather than throughout the whole solution. And it also stabilizes the enzyme chemically by lending physical stability from the solidity of the support. And it also prevents exposure to the heat and pH in the regeneration unit, which also stabilizes the carbonic anhydrase. And stabilizing it makes it last longer, which means you need to replace it less often, which saves you some cost. Now, also in principle, immobilized carbonic anhydrase can exceed the catalytic activity of free carbonic anhydrase. And we'll talk a little bit about that later. And so if you can achieve that, you've also made a better catalyst. However, currently immobilization tends to reduce the activity of the catalyst because it disrupts its structure. We'll talk about that in a few slides. And it also overall just tends to have poorer absorption enhancement, and we'll talk about why that is as well. So I'm not aware of any analyses really comparing the overall techno-economics of these two approaches. And so the first thing is that it would be great to have an analysis of that, which catalyst properties and process parameters, we're talking about absorption rate, enzyme cost, materials cost, the temperature and the pH, economically favor using a free or immobilized CA. And what are the sort of conditions that each has to meet in order to win out over the other? And another analysis would just be, what are the minimum and ideal catalyst properties and the corresponding process parameters for using carbonic anhydrase in DAC? Because if we can make them milder by making the catalyst better, then that might mean the catalyst isn't being destabilized and we can just use free CA, which is simpler. So over the next few slides, I'll talk about how Biotech can improve the performance of both free and immobilized CA, regardless of which path we choose. So we'll start with free CA. It's conceptually the simplest. So, and again, we're still talking about using biocatalysts in DAC. So reducing the cost of CA is the core challenge for using free CA. And that winds up relating to the stability. So the two cost factors in using CA are the stability of the enzyme, which really determines how often you need to replace it. And the other is, of course, just the manufacturing cost. So, so it turns out that as regards stability in harsh conditions, the most powerful protein engineering tools haven't yet been applied to CA, and we have made a lot of progress even without them. So if we consider the most stable published carbonic anhydrase that I was able to find, it impressively has a half-life of 203 days at 60 degrees Celsius, which is incredible for a protein. And it was discovered by isolating one extremophilic carbonic anhydrase. This means it's a carbonic anhydrase from an organism that lives in very hot conditions, and then screening 6 rationally designed variants of that enzyme that produce this success. But metagenomics can identify thousands of variants that we could draw from. Now, metagenomics being a term that refers to collecting DNA samples from communities of organisms in a specific environment, and then analyzing the DNA sequences that come out of them and looking at the different organisms. And so, we can sample DNA from extreme conditions, and we could look at all the different carbonic anhydrases in there. We could screen them, and we could learn from them and use that as we design CAs, rather than doing them one by one, which seems to be what one by one or a few by a few, which seems to be what has mostly been done so far in the literature. And we can also use AI-driven protein design in addition to rational design, because AI-driven design is really revolutionizing protein engineering right now, and so it could be very helpful for producing high-performing, hyper-stable carbonic anhydrases. And so we've discovered a fundable project here, which is applying cutting-edge protein engineering and metagenomics to optimize carbonic anhydrase. Can we use AI-driven protein design to produce a hyper-stable carbonic anhydrase? And can we screen carbonic anhydrase from many thousands or more extremophiles to discover starting points and design rules for engineering carbonic anhydrase? Now, the other cost factor we mentioned is the manufacturing cost of the enzyme. And scaling the manufacture of an enzyme is crucial for removing cost barriers. So enzymes can be extremely expensive to produce, especially at small scale. So in academic labs, it can be like $1,000 a milligram. But then as you scale it up, you gain economy of scale. You also optimize the process, and you can get that cost down to $50 to $500 a kilogram, or even lower, $10 to $30 a kilogram, which is crazy. I mean, the hemp protein powder that I eat is more than $10 a kilogram, and we're talking about $10 a kilogram here for a purified protein. It's totally wild. Now, the company Novozymes has produced a thermostable carbonic anhydrase, which means it's stable at high temperature, and you can get that for $480 a kilogram, which is really impressive. And if that were optimized and scaled further, it could become tenfold cheaper. For example, the proteases that we use in our detergents are about $10 a kilogram. And the reason for that is basically because those are just the most used and highly manufactured enzymes in the world. And if carbonic anhydrase indeed winds up being used ubiquitously in point source capture and direct air capture, it also may be one of the most widely produced enzymes in the world because of what scale those technologies need to reach. And so that means that in the future, the cost of carbonic anhydrase may actually be far less than the cost of the energy and the air contactor that are driving the cost of DAC. So we may not actually care as much about the cost of the enzyme once these technologies are scaled, because they have a very impressive economy of scale. And so that indicates that we might actually care more just about the performance. Now, another detail here, thinking about scaled manufacture, is that there are other useful carbonic anhydrase variants that we might want to use in technologies other than this specific one that has been scaled by Novozymes. And so if we want to use those, then we have to scale them separately. And we should think about, once we recognize the need for that, doing that as quickly as possible. So there's some projects here. Another instance where protein engineers need to work with the materials and process engineers to help everybody. As for the community infrastructure, can we apply financial market shaping instruments to drive down the cost of carbonic anhydrase so that it reaches that low cost as soon as possible? And specifically, this would be for something like the already scaled carbonic anhydrase that Novozymes is using. If $480 a kilogram is gating people, perhaps we can help that chicken and egg problem by incentivizing increased optimization through committing to purchasing the enzyme downstream. And the other thing is, can we apply the cutting edge of biomanufacturing, which is an ongoing technology challenge that actually has received revived attention lately, to make impactful variants of carbonic anhydrase cheaply, so that we can start using the most optimized carbonic anhydrases for specific applications. The other question here is just, how cheap does carbonic anhydrase need to be? It would be great to see a techno-economic analysis telling us when the carbonic anhydrase is cheap enough and cost optimization yields diminishing returns because the cost of the enzyme is just no longer anything close to a dominant cost in the technology. We don't know right now, at least in the published literature, when that happens. So now let's move on to talk about carbonic anhydrase in immobilized CA. Or sorry, let's talk about the use of immobilized CA in direct air capture. So the durability, the activity, and the placement of the immobilized enzyme at the gas solvent interface are the core challenges for using immobilized carbonic anhydrase. So in terms of durability, it has to be durable so that it doesn't leach off the solid support that it's attached to as the solvent continuously flows over it. It has to retain high activity. I mean, the activity is the core of its catalysis, of its function, and it has to retain that function while being chemically anchored, which is a modification to the structure of the protein, which is a subtle challenge to have a protein maintain its function while also chemically modifying it. And the third is that carbonic anhydrase has to be present at the gas solvent interface so that it can access the gas that it's trying to catalyze. And we'll dig into each of these now. So currently, immobilized carbonic anhydrase often trades activity for durability. So on the high activity side, we can make nanoparticles and other materials that have extremely dense carbonic anhydrase that exceeds the solubility limit because it's densely packed. And in this case, we're looking at an image of what they're calling a carbonic anhydrase nanoflower particle, which is like a precipitated composite of carbonic anhydrase with a mineral. And it forms these very high surface area structures with very concentrated catalyst. And this can achieve threefold enhanced activity over free carbonic anhydrase, which is already one of the fastest enzymes known. I actually should have mentioned that earlier, that carbonic anhydrase is one of the fastest enzymes and most efficient enzymes known to humanity. Now on the, oh yes, but this, in addition to being high activity, this kind of structure is very fragile. And so if you expose this to the solvent flows that you get in direct air capture, it's gonna dissolve or break, probably break, and won't be useful for that in that physical context. Now on the other side, we have durable anchoring of proteins, so we can use chemical cross-linking. So this is a covalent bond that anchors the proteins to a solid support. But that can tend to reduce durability because those chemical cross-links can interfere with the structure of the protein. And so that tends to reduce the activity. And so you don't get that super enhanced activity that you get from the high density enzyme. Now it's possible to use specific cross-linking to durably anchor enzyme to durably anchor an enzyme while preserving its activity. So let's look at this more closely. If you use random cross-linking, like has been done in the majority of carbonic anhydrase immobilization to date, you can structurally damage the enzyme because the random cross-link attaches to the enzyme at a random location. Sometimes the enzyme will be in an orientation and structure that allows it to still function, and some of the time it won't. But if you get a little more fancy and a little more specific, and you engineer either the protein or the cross-linking process to specifically anchor the protein at a certain location, you can ensure that the protein is in its correct orientation every time it attaches to the substrate. And that's when you start to be able to get really high density, high functioning catalysts. And so the fundable project that we found here is, can we apply site-specific anchoring techniques to immobilize carbonic anhydrase durably and densely while enhancing activity? And I should say that these site-specific anchoring techniques are already under development. Many of them are already under development in pharma, also in enzyme immobilization and in imaging technologies. And so there is a lot of expertise that could be brought to bear on this problem. Now, the final challenge I'll talk about is placing carbonic anhydrase at the gas-solvent interface. This is crucial. And actually, if you don't have this, you have nothing. Because the specific reaction that we're trying to catalyze here with this enzyme is an interaction between two phases, between gas and water. And so the gas and liquid. And so the catalyst needs to be present where that exchange is happening, which is right at that interface. If the enzyme is too deep into the solvent, like on an immobilized substrate, and the solvent is flowing over it, the carbon dioxide has to diffuse through the solvent before it reaches the catalyst. You basically lost the benefit of the catalyst because diffusion is a relatively slow process. So just another sort of way to look at this is that if the enzyme is close to the interface, you get a relatively lower amount of carbon dioxide right at that interface because it's immediately being catalyzed. And having a low partial pressure of CO2 in that at that interface is going to drive gas into the solid or into the solution. Whereas if you have a higher concentration at the interface, like you have when the catalyst is far from the interface, you just have less of a partial pressure differential and less CO2 going into the solution. That got a little technical there, but suffice to say, you need the enzyme at the interface. How close? Well, you need it about, ideally, within the first 10 microns, according to a modeling analysis that was done by Penders Van Elk. And so that's a really small distance. That's like 1/10 of a hair. And so it's really a big engineering challenge to get a catalyst that close to an interface. And this has been the key limiting factor in immobilization to date. Now, this proximity can be engineered. The key factors are a high surface area of the immobilized substrate and using a very high wetting material so that the liquid so the solvent will wick over the material in a very thin layer. And I want to highlight a really cool example of this by Shen and colleagues in Sonia Salmon's lab at North Carolina State. This was published last year. They're using textiles, cotton fibers, which we know is a very high wetting material. If you think about like a cotton towel, the way it absorbs water, it very uniformly spreads the water over the textile. They're immobilizing carbonic anhydrase on here and then wicking a solvent to flow along the textile, which has very high contact with air. And they were able to achieve a 19-fold absorption enhancement using this technology in the case of point source capture, using a weak base versus compared to the same weak base in an air contactor. So just to be clear, this isn't a 19-fold enhancement over the state of the art. It's a 19-fold enhancement in that weak base. Now, this actually has a lot of room for improvement because the anchoring methods used here are using that random cross-linking, and so we're not fully capturing the catalytic capability of the enzyme yet. And so we've discovered here another fundable project, which is at the interface of protein engineering and materials and process engineering. Can we make a high surface area, very hydrophobic substrate for immobilizing carbonic anhydrase that then enables CO2 absorption to be as fast or faster than free Ca, basically living up to the potential of immobilized carbonic anhydrase. So 2 more fundable projects here that I'll discuss quickly for enabling the community. The first is addressing access to carbonic anhydrase. Many useful variants of carbonic anhydrase exist that are optimal and stable in different conditions of high temperature or high pH or salinity, but these aren't commercially available. The only commercially available ones really are human carbonic anhydrase, bovine cow carbonic anhydrase, and this thermostable carbonic anhydrase from Novozymes. But there are so many more. There are thousands that we might be interested in using. And you can't expect a CDR researcher to synthesize their own carbonic anhydrase. That's like asking someone to write the software to actually write the code for the software that they need to use. It's just a totally different skill set, and you can't expect like a DAC company to have a biolab. And so we need a lab that's willing to synthesize and quality control carbonic anhydrase variants to support bench scale research in the community. And the last project I'll talk about is mapping the patent landscape for biocatalysts in DAC and DOC. Almost no work on using biocatalysts in DAC and DOC has been done, but there has been substantial work in point source capture. And so what do the opportunities look like for using carbonic anhydrase in a set of technologies that are adjacent to a field where it's already being used? And so that would be actually not a TEA, that would just be a study, but can we help newcomers understand the IP landscape in the space of biocatalysts for CDR through a short report on the topic? So I'll quickly summarize what we've talked about. I know I've gone a little over time because I wanted to leave time for questions. So we've discovered that biocatalysts can meaningfully reduce, in principle, the cost and energy of direct air capture and direct ocean capture through reducing regeneration energy and reducing contactor size. And in DOC, saving the electrodialysis energy and the electrodialyzer cost. We found that we need clear engineering targets to help people work towards specific goals in their research. And we need protein engineers to work with materials and process engineers on these problems. We found 14 fundable projects. I'll just summarize those now. In DAC, technoeconomics analysis, tell us the minimum and ideal performance criteria and when to use free or immobilized CA. Engineering, basically, can we use the cutting edge of protein engineering and screening, metagenomics, to optimize the performance of carbonic anhydrase for DAC? In DOC, again, we need to know the minimum and ideal performance criteria and process designs for using catalysts in DOC. That's an analysis. And then we also need to make biocatalysts and membranes that maximally leverage each other's strengths. We need a lab to synthesize and quality control the variants to support bench scale research in the community. And we need to drive down the cost of carbonic anhydrase and its variants as fast as possible, specifically for applications we know are important and that we want to scale. And then finally, can we help newcomers understand the IP in the space so they can see where the opportunities are? Now, if you want to discuss any of these proposed projects, like I said, we're looking to enable them. We need everybody from scientists to governments participating in accelerating this kind of research. Please reach out to me at paul@homeworld.bio. I'd love to talk. So at this point, I just want to say thank you again to my funder, Additional Ventures, for enabling me to do this work, and for Innovative Genomics Institute at UC Berkeley for hosting me. And now thank you so much for your interest and for coming. And I'll take any questions you have now. I think.
All right, very, very good. So we've been having banter in the Q&A, and so I can answer. I'm going to ask a couple questions on behalf of the people who were commenting. So the first one was going all the way back, and there's a question about pH. So the question was, what's the mechanism that carbonic anhydrase uses to extract CO2? But I think more specifically, because that can be pointed to in the report, the question was, does it cause a shift in pH?
Yeah, so anytime that CO2 dissolves into a solution, it acidifies the solution. And anytime it comes out of the solution, it basifies the solution. And the catalyst is just making that process happen faster. And it'll make it happen in whichever direction is thermodynamically favorable, just faster.
Got it. And then there's another question about MRV. And so is there anything different about MRV for a carbonic anhydrase catalyzed solution versus just a normal DAC or DOC setup?
So MRV stands for measurement.
Reporting.
The R stands for measurement and verification of to confirm carbon removal. In direct air capture is the easiest technology to do MRV. And so this would just enable the technology that's easiest to do MRV. DOC, you can know how much carbon you've captured, but it's harder to confirm that has actually come out of the air to re-equilibrate with the ocean. Yeah, I would say the answer is no, this doesn't affect MRV. But it does improve technologies that have very good MRV.
Got it. And so we've got about 90 seconds left to end on time. So we have one more question that has already been answered, and then we're going to go through the latest 3 that just came in. There's a question about how would all of this work for a Climeworks type DAC, where you have a solid support functionalized with a liquid solvent. In Climeworks, it's amines.
Yeah, great question. So the liquid solvent, my understanding of solid supports of solid sorbents is weaker, but my concept for them is that you have a solid atop which there's a very thin film of liquid that CO2 has to enter in order to come into contact with the solid. I'm actually not sure if that transfer of CO2 into that thin film is the rate limiting step, but if it is, then carbonic anhydrase can help. And you would need some sort of composite of the amine and the carbonic anhydrase in order to do that, which might be challenging given that the lifetime of the carbonic anhydrase might be less than the solid sorbent. I'm actually not quite sure, but that is, yeah, you would basically need to find a way to confine the enzyme within that very thin film on top of the solid.
Okay, great. And then there's one last question, and it seems to be very broad. So, which
I don't
is,
mind staying like an extra couple minutes. I mean, people might drop off, but if folks do want to hear a few more questions, I'm happy to answer them. And folks can feel free to drop.
well, thank you, everybody. In that case, thank you, everybody, for coming.
Yeah, thank you for coming.
For those that do want to drop off now.
Yeah, thank you so much for coming. I'm sorry that I went a few minutes over in my talk, didn't leave as much time for questions. So if you have other things to run to, I will not be offended. But yeah, thanks for coming. And again, if you wanna work on any of that stuff or contribute to it, reach out to me, paul@homeworld.bio. And I'm happy to talk about a few more questions now.
Okay, great. So then, as people decide to stay, it looks like actually a lot of people are staying. That's wonderful. There's a question of, do you have a personal view if carbonic anhydrase will be more feasible in DAC versus DOC?
I think the application, will it be more feasible in one versus the other? I'm not sure. I think in DOC, it's likely that the first application is going to not be a carbonic anhydrase, but rather a mimic, a small molecule mimic of the same that catalyzes the same reaction. It's more stable. The method for implementing it in DOC, at least conceptually, is clearer. And so in that sense, it may happen for DOC sooner. Really, you just have to figure out a way to economically immobilize a catalyst on the inside of the hollow fiber membrane, and make the catalyst cheap enough and stable enough to work for you. That's a pretty well defined problem.
Okay, great. And then We have a question from Simon, which is, can you talk a bit about how the higher level economics of CDR relates to the gains in efficiency? Is there a threshold of efficiency which opens up demand for Docker DAC?
That's a great question. So and it has a few parts. So the first thing is that people have generally said that $100 a ton for carbon dioxide removal is a target that would make it economically favorable. Now, whether that's true or not and where that number comes from is a separate question. We might be just saying $100 a ton because 100 is a round number. And the social cost of carbon is estimated to be higher than that. The examples I gave showed that sort of in the best case scenario, based on like a really light back of the envelope, is that we could reduce in the case of dock, this could be like 40% reduction in cost. Again, that's a very best case scenario that you should not take my word for. We need an analysis to actually answer that question. And for direct air and in point source capture, that they found that carbonic anhydrase a real techno-economic analysis found a cost reduction of 31%. And so if the other components of DAC and DOC can be optimized to the point where a 30% reduction or something like that using an optimized catalyst actually brings you over that threshold, then you're going over the threshold. So I don't want to say whether this technology in particular can bring you over that threshold, because it really depends on the technology as a whole. And I've been trying to do my best not to make promises like that throughout my road mapping effort, and instead say, this could contribute X% roughly of a optimization. That was a long answer, but.
Great. So we have a question from another anonymous attendee, which is that right now enzymes are being discussed as adjuvants or accelerators of current Docker DAC conceptualizations. However, enzymes in nature have many functions, presenting the attractive possibility of replacing current DAC or DOC processes. Is that a topic of discussion, or is it such a strategy too undetermined for people to explore?
I think you could explore it. I actually haven't explored this that much because it seems hard, but sort of wish that I had dug deeper into that. But I think the question you're talking about is like, for example, we have all these proteins that fix carbon. So can we absorb carbon into a solution and then use an enzyme to fix it into some other molecule, which is sort of like the technologies that people are building to use captured carbon to make fuels or materials or other useful stuff. So using free enzymes to do that process is a challenge already to make cost-wise to make commodity chemicals. And so it's also, I think, a challenge for capturing carbon on its own. Now, an interesting application of carbon fixation would be just to fix that carbon into biomass after absorbing it into a solution, in which case you're imagining like a liquid containing a bunch of algae that are photosynthesizing and continuously pulling carbon into the solution, which is an idea that I'm interested in and have been looking at in some detail. And I think that actually could be useful, but that would be an organism doing it, not a cell-free protein solution. But in short, I think it's worth looking into the cell-free protein solutions.
And there was one paper, I think, in 2021 about cell-free starch synthesis using atmospheric CO2. And I think it was a very interesting science paper, but it becomes, there's more a demonstration of potential rather than something that would actually scale, because I think it was using very exotic catalysts and things like that. So is there any filtering ahead of the hollow fibers in DOC? So this is another anonymous attendee. Is there any filtering ahead of the hollow fibers in DOC? And if not, how is the biological density of seawater expected to reduce performance or half-life of functional enzymes?
Okay, so the first question is there filtering? And the answer is yes. And that doesn't really have to do with I mean, that has to happen regardless of whether you are using a catalyst, because you're passing the seawater through a hollow fiber membrane, which is a very thin tube. And if you're putting a bunch of biological material through that, then those membranes are going to foul and clog, and they're very expensive. And so there is a pre-filtering that I think is like, I wish I knew this off the top of my head, but it's a pretty fine filter. It's like I think it's like 100 micron or maybe smaller filter. Should be smaller, otherwise you'd be letting cells through.
Great.
Oh, and the second half of that, I think, was about biofouling. Biofouling is an interesting question for this whole set of technologies, which I did discuss in the written roadmap. And it's basically something that needs to be accounted for and needs to be mapped out with some kind of analysis. Like, what sort of impact do we think biofouling is going to have on the cost? Because that doesn't seem to be addressed in most of the papers that I could find. But anecdotally, from some folks developing this kind of technology, I've been told that, at least at bench scale in the environment that they were using, biofouling was a challenge. Biofouling being organisms eating the carbonic anhydrase and growing in your solution.
Great. And then I was just answering a question saying that carbonic anhydrase is temperature and pH sensitive. Do you think efficiency will vary over time?
So the enzyme will degrade over time, and temperature and pH are known to damage proteins. And so a core challenge of developing proteins for the use in industrial processes is having them tolerate the temperature and pH of those processes. And that's where we have to apply biological search methods like sequencing and metagenomics to look at existing proteins that are in extremophiles. These are organisms that are very happy at hydrothermal vents that are above 100 degrees C, and they're growing there, and they are full of proteins that are stable at that temperature. And there's also organisms growing in extreme high pH and extreme low pH. And we can start with the enzymes that those organisms use, which is what people have been doing. And then on top of that, we can apply techniques in protein engineering to make those even better. And again, that's what people have been doing so far. Now, my argument here is that using the newer wave of technologies that we have now in the ongoing revolution in protein engineering, it's likely that we can beat the stability and performance that has been obtained to date, which already is showing promise.
Great. So we have two more questions. And actually, it's a perfect follow-up to what you just said, which is that what cutting-edge biomanufacturing solutions do you have in mind to drive down the cost of carbonic anhydrase variants? How do you imagine enabling unlocks for these manufacturing techniques?
That's a good question, and I'll admit that that's a topic that I have less thoughtful things to say. Although I definitely would love input from others. One thing I'll say is there is a financial instrument that you can apply to this sort of thing, is something called a volume guarantee, which is which is sort of a market pull technique, where if you have some material that has an economy of scale, meaning that if you make more of it, it will become cheaper. You can, and if you're confident enough that once it becomes cheaper, you're going to want to use it, then you can make essentially get like a consortium of companies or some large buyers to commit to purchasing that material at a lower price at a large volume in the future, which incentivizes people to build that scaling and build the new infrastructure to produce at scale. And that can just accelerate the scaling and drive down the cost of a material. In the case of variants that are currently not produced at scale, I think the first step would be to produce them at all in a way that makes them available to researchers. And then once you've identified that that one of those proteins is of high enough interest that you want to scale it, then I think we need to apply biomanufacturing technologies to that, and the best that we possibly have. I mean, I don't have actually a nuanced understanding of the most cutting edge emerging biomanufacturing technologies, but I know that there's innovation there, and I would say that for folks who are developing new biomanufacturing technologies, consider using carbonic anhydrase variants as a case study, or as a pilot, or as a first opportunity in the space, because it could have a profound impact on carbon dioxide removal.
Great. A quick question is, hollow fiber, is it working on the adsorption or absorption principle?
Hollow fiber membranes, the way that they're working is, actually, I can just go back to the slide. Wow, it's way back. I had a lot of slides. I must have passed it. Oh, yeah. Okay, we made it. So the way the hollow fiber membrane works is it's composed, it has two layers, at least two. Some hollow fiber membranes can get really fancy. But you have this selective layer, which allows only certain chemicals to pass through. And then you have this porous layer, which is more of a support structure. And in the case of DOC, the seawater passes through the tube, and water can't pass this selective membrane, and CO2 can, and so the CO2 comes out, and that is the reverse of absorption. It would be an absorption, a deabsorption. What is the word for that? Definitely not adsorption. Definitely not adsorption. Adsorption being the binding of like a molecule or something to a surface or another molecule. Okay, I hope I answered that well.
I think so, yeah. So I think there's one question at the top, which I think is something that people might misunderstand. So I'm going to end which is that carbonic anhydrase is there to catalyze reactions and speed them up, and they're not necessarily useful when it's not the limit, like the CO2 hydration is not the limiting factor. So that, anyway, that's my pre-answer to set up the question for your actual answer, which is that,
oh yeah,
can you have a new high concentration carbonic anhydrase producing algae so that we can push air water through that system to increase today's performance in some sort of future photobioreactor?
Wait, so first you made a comment and then a question?
Yeah, basically, like, I think there's a general question. There's a couple questions in the chat
I want to focus on your comment for a sec, because it's something that I wish I had stressed, because it is a common misunderstanding. So carbonic anhydrase is an enzyme. It's a catalyst, really, that that speeds up a specific reaction, which is the interconversion of CO2 with bicarbonate. And under conditions where you have high exposure of CO2 to the liquid, so you have highly accessible CO2, and you are not limited by diffusion, this reaction is the rate limiting step. And so carbonic anhydrase has evolved specifically for that. It doesn't accelerate any mass transfer processes. It doesn't use up energy to make anything faster. It just catalyzes an existing reaction. And so if this specific reaction is not what's limiting your mass transfer, so for example, if you don't have enough access to CO2, then the enzyme is not useful. And actually, a perfect example of that we talked about is in the case of the immobilized carbonic anhydrase, where if oh wow, so many slides. If the enzyme is not close enough to the source of CO2, then it's the diffusion of CO2 that is the limiting rate. The catalyst can be infinitely fast, and it's not going to do anything for you. And so I think in general, these are two factors that people need to remember. One is carbonic anhydrase is not a pump that actively increases the transfer of CO2 using some sort of energy. It's just a lubricant to make thermodynamically favorable processes happen faster in terms of that specific reaction. And the other one is that it's only useful if that specific reaction is what's limiting your rate. And so if you're considering using carbonic anhydrase, always ask yourself, is hydration of CO2 interconversion with bicarbonates the rate limiting step? Because I see people considering carbonic anhydrase frequently in situations where that's not the limiting step. That's a long spiel, but I think it's really important.
Yeah. So then is there any reason to think that making more CA in an algae to make it grow faster would work?
Okay, so if an algae is making more CA to grow faster, the way that would work is if CO2 turning into bicarbonates is what's limiting the uptake of CO2 into the organism, and the organism has enough light coming in to fix it faster. I think in certain cases, yes, that would be possible. And so there are applications, like there's point source applications where people want to use yeah, I won't go into it, but I think the answer is yes. And actually, there's a paper on this. If you look up like carbonic anhydrase fixed to alginate beads, I think it is, using glutaraldehyde, if you like look at those terms, to accelerate the transfer of CO2 into a pond for algae growth. That's a pretty cool demonstration. It's an ACS chemistry, I think, ACS something, ACS synthetic biology.
Got it. So I would say we probably have about 10 more minutes and we wrap it up. So we've got a few questions to go through.
Hey, 10 minutes. Let's go.
So are there advantages, and I think you've already you answered this, but we should just go through it again for completeness. It said, are there advantages to using, oh, I'm sorry, wrong question. I don't think you've answered this. Are there advantages to using non-aqueous but organic solvents in DOC or DAC? Are there enzymes that retain their function in such a system? So basically,
yes.
Can you do carbonic anhydrase in a solid system?
You could. So I think the question was asking, could you do it in a solid or liquid system that just isn't aqueous? Like it's not a water solution. So enzymes can work in solutions like that. Most enzymes don't, but enzymes can work, and you can engineer them to work in those conditions. The mechanism that carbonic anhydrase uses is catalyzing the reaction of hydroxide or water with CO2. And so for CO2 to become hydrated, it does need some water present, or at least hydroxide. And so the chemistry of that scenario would be a little subtle, but probably what's most likely is you have like some small amount of water. So I think in principle, yes, it could be good. If there's a specific use case where, again, that specific reaction, interconversion of CO2 with bicarbonates, is a limiting factor, in the context of a non-aqueous solution, then maybe carbonic anhydrase could help, and at that point it'd be worth asking.
Great. All right, the last question, and it's a topic I really like. What would it take for DAC systems based on artificial enzymes to outcompete the organic or the original carbonic anhydrase?
Great question. So the question is, at what point do catalysts that are not basically non-protein catalysts, artificial materials, to outperform the carbonic anhydrase? So I think in the abstract, we don't really care whether it's carbonic anhydrase, we just want a catalyst. Now, it turns out that carbonic anhydrase is, by orders of magnitude, the best catalyst for this interconversion of CO2 and bicarbonate, which tends to be the rate-limiting step in CO2 absorption for optimized systems. Now, carbonic anhydrase has a few weaknesses. So I guess the first thing to say is, if you could make an artificial catalyst that performs equally well to carbonic anhydrase, then there might be scenarios where you'd want to use it, especially if it's cheaper. Carbonic anhydrase is a protein, so it can degrade. Some of these artificial versions can be more stable in particular conditions, so that could be an advantage. Also, sometimes you don't need the full catalytic capability of carbonic anhydrase. And so we actually discussed an instance of that, which is in the context of DOC. We expect the permeability of hollow fiber membranes to limit the flux of CO2 across the membrane more than we expect the permeability of the membrane to limit how fast that reaction can go. So the full catalytic capability of the carbonic anhydrase isn't used there. And so in situations where like you can't, where like having an extremely efficient catalyst isn't useful, because once you have a good enough catalyst, like some other factor limits you, then you might ask, well, can we use just a less efficient but more stable, or more some other property that's advantageous? So it's really a case-by-case basis.
Great. Okay, well, that is all the questions. And I think Paul, you did a really amazing job. And I love that there are still 21 people who have been hanging out listening to this. So if people want to continue the conversation, Paul, how do they find you?
You can email me at paul@homeworld.bio. That's probably the easiest way. Or find me on LinkedIn, or however else you find people.
Awesome. All right. Well, thank you very much. Really amazing work and fantastic questions from the audience. This was really, really cool.